All right. So I'm Amina Fine. I know that. I know the Gilliams, obviously from our natural history study. We appreciate you being here. Where did you all come from again? That's right. Okay, good. Yeah. So, yeah, I'll give a little bit of background. You might not even know, I guess. So I'm faculty at Kennedy Krieger, and I'm assistant professor of neurology. Been in that position for 2 years. I guess I actually been at Kennedy Krieger and Hopkins since 2013 for fellowship in pediatrics and neurodevelopmental disabilities, which is like child neurology, adult neurology, and developmental peds. So, our LBSL program started in 2015, I believe yeah, when, the McGinn family of the LBSL Foundation or Cure for Ellie first approached our boss, Dr. Ali Fatimi, about getting a program started in LBSL. And then I got very lucky because I was a first year fellow. I had just sort of switched over from Gen Peds at Hopkins to doing the neurodevelopmental disabilities part at Kennedy. And I was 6 months in and looking to do research. And so Ali introduced the idea of LBSL, and he's like, we're trying to get this program essentially started from the ground and need somebody to focus on clinical history, natural history. So, it was a no-brainer for me to join the team at that time. So that was like early 2016. And so essentially, Christina and I have been working over the last few years kind of on our respective arms. So me more on, the clinical science and natural history, and her more on the basic science program. And doing more work together recently to try to, as we get closer, we hope to developing potential treatments and outcome measures to use in clinical trials in LBSL. So pretty much, I think we can do introductions, and then especially since we're such a small group, maybe take questions or things that you might want to talk about, just things that might be top of mind. We can certainly address those. I have some slides so we could go through more formally, or maybe as ideas come up in discussion, I can focus on those, at the time. Okay, right.
Hi, everyone. I'm Christina Nemeth-Mertz. I've met most of you before. I remember from our last in-person meeting a couple years ago. So I finished my graduate, my PhD in 2014 down in Atlanta at Emory University, and was looking for a postdoc and came to Kennedy Krieger to do some animal work. Basically, I worked with a neonatal hypoxia model with an investigator at Kennedy Krieger, and eventually switched over to working with Dr. Fatimi. And so at the time he had some ALD mouse models, and I was working on that, and we kind of worked to develop this mouse model for LBSL, which I think some of you are familiar with. So that was sort of my start with Ali, and we eventually sort of broadened the program to include the patient cell lines, which has been sort of our main bread and butter right now, and our sort of biggest focus in research at the moment. So I help to run the basic science lab. So we have a small lab of about 4 people. Dr. Manu Amanat is sitting back there. So he's working a lot with the patient cells right now. And and then we have 2 other technicians and one student who just left, who did some really nice work on single cell RNA-seq. And I have slides on all this stuff, so we can talk about whatever anybody wants to talk about. Yeah, so that's basically what we're doing. Like Amina said, we're sort of trying to work on developing a couple different therapeutic arms. So one of them is this antisense oligonucleotides, which Manu is working on, and another is a AAV9 virus, which is aiming to sort of replace the defective DARS2 protein to sort of introduce healthy protein into the cells in the mice. So these are sort of what we're working on. And eventually the goal is that Amina's leg and my leg will sort of combine, and we can take some of these therapeutics into the clinical trials and things like that. So similar to Amina, I prepared several slides because I wasn't really sure what to expect with our crowd. So anything that anybody wants to talk about, we can do an update on research or talk about sort of more of the nuts and bolts about how some of these things work. So happy to let everyone guide how this goes, and sort of looking forward to updating everybody and making sure that everyone has a really clear understanding of what we do and why we do it, and hopefully the next steps moving forward. So should we pass it around to people? Okay. You just bring, you don't have to say much.
I'm Stephanie Gilliam,
Dan Gilliam.
and we have 2 boys with LBSL, ages 6 and 3, and we're just trying to find the best care and treatment for them.
And participate safely in the natural history study.
Hi, Donna Gilliam. I'm Dan's mom and grandmother to Dean and Dalton. Thanks for having us.
And I'm Nancy Schuwerk, and I'm Stephanie's mom and Dean and Dalton's mama.
Hi, I'm Kim Hollinsworth. I'm a nurse at Kennedy Krieger, working in leukodystrophies.
I'm Dan Amos. I am the lead research coordinator for Amina's LBSL natural history study.
Hi everyone, I'm Emmanuel Monat. I'm the postdoc fellow in Johns Hopkins. I was graduated MD from Iran, and I'm working with Amina, Dr. Mers, and Dr. Fatimi since October. Thanks.
I'm Christy DeMarco. I'm a ULF board member and also a patient of a leukodystrophy, not LBSL, but Refsum's disease. And I'm really always interested in, we're really early on as well. And natural history studies are very interesting to me. So I'd be welcome to listen in and learn from all of you. So.
Do we have anybody online yet? One person. Perfect.
Okay, so yeah, so I guess we could I mean, if there are any like particular questions you all have, we can start there, and then I think once that's done, maybe I can start going through some of the natural history slides. Yeah, okay. No pressure.
I'm curious about the natural
Yeah.
history, and then also like is what you were talking about, like a gene therapy, is that something that I guess I'm interested in hearing more about?
Okay. Yeah, so
I don't have any actual, well, I feel like you mentioned the dark protein.
yeah.
So what happens to people that have already had some regression if they undergo gene therapy? Is it hopeful that we can have generation of new cells, new mitochondria? I mean, how does that impact?
Right. So that's a good question. And I think that's something that, should I answer this now or just, yeah. So,
Yeah, I think there's like some general broad
Yeah.
strokes we can give about.
so, I mean, that's a really good question. And I think that's something that we're not going to know until we sort of get there. The idea is that, so obviously the patients have some functional DARS2 because if they didn't, I think it would be a much worse situation. So the idea to introduce more healthy protein to sort of, shift that ratio of defective to healthy protein to sort of, turn it in the right direction. Whether there's the ability for regeneration or repair, we don't know. There would certainly, hopefully, be a sort of, halting the disease progression. I think that would be sort of best case scenario. Whether there's regeneration, again, it sort of depends on what DARS2 is actually doing. In my mind, we know that DARS2 has a role in sort of translation, making proteins within the mitochondria. And there certainly is mitochondrial deficits in a lot of these cells that we see. I sort of believe that DARS2 is doing other functions. And so depending on what these other roles are, if we can replenish that, I feel like there would be a fairly strong, there would be a pretty good chance of, of halting that progression and being fairly functional after that. Whether that repairs, some of the issues, some of the damage that's been done, I don't really know. That's sort of a complicated response, but one of the good things I think about LBSL compared to some of the other leukodystrophies is the progression is pretty slow, compared to ALD or something like that. So I think there's a decent enough window in there that you can sort of make some headway with a treatment that maybe some other kids in ALD wouldn't, be able to get. So the question was just, I guess, to review for our listeners online was, in the event that there's a gene therapy that's able to introduce healthy DARS2 protein into patients, whether or not that would be able to sort of regenerate damage that's been done to nerve cells or myelination and things like that, or whether we could halt progression. And so this is still work that we're looking at. And I have a lot of data. Some of it, I think some of you have seen before. And I didn't prepare any slides for the AAV9 work and for the ASOs because it is really new. And really all we've done with that is to test it in a single cell line, sort of multiple times under multiple iterations. And I can talk about it a little bit more later, but both of these approaches that we've taken have shown promise in these cell lines that, we can repair some deficit that these cells had at baseline. The next step would be to do it in multiple patient lines, to do it under multiple conditions. And then we know that we can sort of improve the ability of these cells to make a proper DARS2 transcript, which would result in a healthy protein. But the next question is to see if we can improve the function of these cells. So if we put these cells through a test where they have action potentials or sort of conduct electricity, or if we put them in a test where it measures their mitochondrial function, can we make those cells behave like a control cell line? And that's something that we're hopefully going to be doing in the next, month or two, something like that. So these studies, before we present anything and before we really make any conclusions, we want to do it many, many times in many different cell lines. And we recently were able to get isogenic controls. So what we did is we actually took some of our patient cells, including your sons, and we sent them to a company, and they genetically repair the mutations in those cell lines and send it back to us. And that way, the only difference between those controls, we call them controls now, and the original cells is the repair of that single mutation. So every other part of, the genome was the same. So we know that if we correct that one problem and the cells behave differently, then in fact, those mutations were the cause of the deficits that we see in the cells. So I think having those new control lines is a really powerful tool. And it's, if we submit papers or if we submit grants, that's really what people are looking for, is that comparison. So I think it really strengthens our argument and will make our data stronger moving forward to papers and the NIH and, the FDA and stuff like that. So I think that's a really important, some important progress that we've made recently. Yeah.
So I'm going to put you all on the hot seat because since we're such a small group, we did introductions.
Hi, my name is Hemmo. I'm a PhD student from the Netherlands, from Amsterdam at the Medical University Center. And together with Mark Engle, who's my supervisor, we see patients with LBSL in clinic. And we have like a standardized way to see them with MRI, blood tests, stuff like that. So it's good to be here.
No, thanks.
Yeah, so that was it.
Okay. So yes, I'll just go on and get started talking a little bit with you guys. Let me just get warmed up here again. Okay. Great. Okay. And I should also say that, in 2019, the last time that we had an LBSL meeting, Mike McGinn was the only one there. So we're already a tenfold larger audience than we were 3 years ago. So yeah, so just happy you all are here. And like I said, this is informal, and so just feel free to, to pause me, to interrupt, to ask questions. That's totally fine. Or for me to give any more clarification. Yep. Okay. So then, all right, so I'm just going to give an overview of, what we learned over the last several years. And so, first of all, just for everybody's sake, because I think a couple people are less familiar with LBSL, reviewing the diagnostic criteria. Hi, and there's Dr. Fatimi. Just giving a brief review of what we know from the LBSL clinical literature overall, and then an overview of our ongoing LBSL natural history study at Kennedy Krieger. And then we'll talk a little bit about the newer partnerships that we have actually with Mark England, who is here and, in recruiting patients in Amsterdam. And then, brand new really is getting a partnership started with a group in Finland at University of Helsinki to involve more pediatric patients in the natural history study. Okay. So LBSL, leukoencephalopathy with brainstem and spinal cord involvement and lactate elevation. So there are three major hallmarks of disease in LBSL. So we know the DARS2 mutations. I'll talk more about that in a minute, but they're inherited in autosomal recessive manner, and they encode for mitochondrial aspartyl tRNA synthetase, which is important for translation of proteins in the mitochondria, especially that are critical for respiratory chain complex function. There are the MRI brain signal abnormalities, and that was really how Mario van der Nap first described LBSL by pattern recognition of these brain changes. So, really changes throughout the neuroaxis in the cerebral white matter, the cerebellum, the brainstem, and the spinal cord tracts. And then this isn't necessary for the diagnosis, but there is most of the time increased lactate on MR spectroscopy in those areas where there's there are white matter changes. And then in terms of clinical characteristics, so, I'll talk about some caveats or some exceptions to this. LBSL was really only described about 20 years ago, so we're still understanding more. The clinical spectrum is still widening in terms of what it looks like, but we know you can have onset in infancy, childhood, or adulthood. Most of the time it's in childhood, more like school age, and then pretty much universally patients have ataxia or unsteady walking, spasticity, and problems with the dorsal column of the spinal cord with a sensory issue I'll talk more about. And as I mentioned, it's really a spectrum from mild to severe disease based on a few factors. So again, the inheritance pattern is autosomal recessive, so basically simply meaning that a single gene variant can lead to disease. So, parents who have one of the DARS2 variants, to our knowledge, are not clinically affected. It really takes two inherited from both of the parents. And then, so as I mentioned earlier, DARS2 encodes, let's see, you can kind of see that, mitochondrial aspartyl tRNA synthetase. So this is the mitochondria, and you can see, so aspartate, one of the 20 amino acids, and here is the tRNA, and so the DARS2 enzyme basically joins those. And this is just important for translation of respiratory chain complex proteins and proper mitochondrial function. And then, so there are not many patients who have the same, there are small groups of patients who have the exact same mutations. There are probably at least 70 different unique mutations that we know of, but one commonality is that 95% of patients, and that really holds over the last 20 years, have a leaky splice site mutation, an intron 2 of the DARS2 gene. So remember, the exons are the part of the gene that codes for protein, and so when there's a mutation in intron 2, that's upstream of exon 3. Exon 3 is like not included in the message or in the gene transcript. And so you basically, in that case, there's a problem with the protein or it's not made. However, it's leaky. So sometimes exon 3 is included correctly and you have, a functional DARS2 protein. So there's a reduction, but not a complete absence of the protein and reduced enzyme activity, which is a good thing. Okay. So just briefly, in terms of what the MRI brain and spinal cord look like. So these are two T2 weighted MRI images. So the kind of the bottom line here, if you can see, the white matter is dark, like almost black. And so kind of where you ever see this sort of fuzzy, bright patches, this is the white matter that is that's abnormal. So it should be looking dark like the rest. The really bright is this cerebral spinal fluid. So this is just to demonstrate how many parts of this is a long white matter tract disease. So everywhere from the cortex through parts of the brainstem, the cerebellum, and the spinal cord tends to be affected in all of those areas in LBSL. And we can come back to that a little bit more later. And as I mentioned, there's often a lactate peak, even though that's not necessary. And so the consequence of this is, so really kind of the major issues with LBSL is motor impairment, because these white matter tracts are really important for voluntary motor control and fine tuning of motor control, especially with the cerebellum. And then, the dorsal columns and the spinal cord, that's affected throughout, and that's really important for sensory input. So sort of recognizing, body position in space, vibration sense, et cetera. And so the unsteadiness that LBSL patients experience is, because of the cerebellum, but also a lot because of what's going on in the spinal cord. Okay. So Dr. Mertz and I published this review article back in 2019, and it was about several of the mitochondrial aminoacyltRNA synthetase disorders, because there are a few that affect the brain and the spinal cord and have neurodevelopmental implications for patients. But this was primarily focused on LBSL. At the time, there were 128 patients, and I basically updated the tables from the paper on the next few slides with a few other cases that have been published since then. So essentially, I think this still holds pretty true, in terms of the mean age of onset. It's in childhood, 8 years. And actually the only thing that's incorrect here is that really now we know that patients have symptoms from birth, starting at birth, and as late onset of symptoms as 40 years. The rate of disease progression, I would say, classically in LBSL for most patients is slow and gradual, rather than more of a rapid decline. And I'll give the caveats there for patients who are affected very early in life. Risk of disability, kind of the major thing we know there is that greater disability is associated with earlier age of onset. And risk of mortality, thankfully tends to be low, but it is increased for infantile onset, or even I'll talk about antenatal onset, where we have evidence, even from in utero or before birth, that there's changes in the brain. And so sometimes those children are actually born with significant LBSL symptoms already. Sorry, I'll pause. Are there any questions so far? I have to remember to to stop. Okay, so alrighty. So just a summary of clinical signs. So again, this is just a bit of an update from the paper several years ago. So there shouldn't be any sex predominance with LBSL. It's autosomal recessive. We were close to 50%. If you look at the literature overall, 132 patients. Cognitive impairment or learning problems in school are in about 30%. And really, I have these stars because, the most common problems really are motor and gait impairment, and 93%. So ataxia, spasticity, weakness, atrophy or wearing of away of the muscles. And that's really primarily affecting the lower body, the legs. And then the sensory changes are also quite common because of the changes in the dorsal column of the spinal cord. And that also contributes to the poor balance and unsteady gait and falling. So otherwise I talked about the cognitive and learning issues. Seizures, thankfully, there's a low percentage, and that's held. 1 to 2% is more common in the general population. So it's more than the general population, but a lot less than in other leukodystrophies. And then things like bladder incontinence or having any visual or auditory problems is also very rare. Mm-hmm. Yes. Yeah. And absolutely. And I'll talk a little bit more about that shortly. Good question. Hi. Okay. All right. So then just to summarize the MRI features, and kind of really no surprises here. I've already talked about how this is a long white matter tract disorder. So symmetry, I guess that's one helpful clue. A lot of leukodystrophies do have symmetric presentation. That just means basically you see changes on the right and the left side of the brain. We're not really expecting that there should be an imbalance there. In terms of the white matter signal abnormalities, really 100% will have cortical white matter involvement. 100% white matter involvement around the ventricles and subcortical areas. The corpus callosum, which is that, that big, myelinated tract connecting the left and the right hemispheres, is often involved. And the pyramidal tracts, throughout and leading into the spinal cord are also almost universally affected. The brainstem as well, especially the medullary tract, the cerebellum, very commonly involved, 97%. We don't usually see basal ganglia, thalamus, sort of other areas there. And there can be, so one thing I wanted to point out is that, well, it's tough because I feel like I know that we don't have a lot of, there's not out there in the literature that's looking at repeated time points at MRIs and LBSL. So, and we don't, unless there's a specific reason, often in leukodystrophies, we're not recommending, to get imaging on a regular repeated basis. But as part of our natural history study, a next phase is to be getting MRIs at baseline and hopefully repeatedly too. So we have a sense of like what those changes are like. So I'll say that at least with most of the MRIs in literature that were just at one point in time, there's not a lot of atrophy or loss of tissue, which is also, I think, a good thing. And then, as I mentioned, having elevated lactate isn't necessary, but it's common at 82%. Okay. So, I've already mentioned this, but I think it's kind of really an important point. So walking is, I imagine, something that's probably really important to patients and to families, developing the ability to walk and then hopefully maintaining that as long into life as possible. And so what we know from the largest cohort of LBSL patients that have been followed serially, and this is group work by Dr. Vandernap's group, that 50% of patients they followed did require a walking aid by adulthood, so relatively high number there, and 13% eventually became fully wheelchair dependent. And in terms of kind of what helped to predict that, it's you can't see the fine numbers with these survival curves, but essentially they looked at patients with age of onset younger than 18 months of age and older than 18 months of age. And those patients who had onset of symptoms before 18 months had the steeper decline in terms of, or higher probability of losing walking without support and becoming wheelchair dependent. So that's just an important thing to keep in mind. And again, the spectrum is still widening. So Dr. Vandernap's group published just in 2021 a case series of 15 patients that, sort of again, looking at the MRI patterns, there was just something unusual that they saw, sort of not that classic picture that I already described. So essentially, so these patients had exome sequencing to identify the DARS2 variant, and then they looked at the MRIs, and it sort of broke down into two different groups. The first group had this very early severe cerebral atrophy and hypoplasia. So I had told you that most of the time there's not atrophy. In this case, in those 9 patients, they saw a lot of wearing away or loss of tissue, loss of brain volume. And so those patients really had a severe picture. They had microcephaly, or they had small heads from birth. They had symptoms from birth. They had not only like loss or slow to develop milestones, but some just didn't actually occur. So arrested development. And then let's see, so they're very severely affected. And then the second group here had white matter abnormalities, but it wasn't throughout the long tracts. And there were 6 patients there. So they looked more severe than classic LBSL, but less worse off than the first group. And then I'll talk a little bit more about the specific characteristics. But when they looked at when they looked at the DARS2 variants that those 2 groups of patients had, and they looked at yeast homologs basically to see what the function was, and they noted that in group 1, those types of mutations really severely impacted mitochondrial function. So it looked like that was sort of the link there. And this might be sort of hard to read, but I think the points here, so we have group 1, and so these patients on presentation, almost 80% had microcephaly. On their later examination, I guess at the time that the paper was written, 100% had microcephaly. let's see, almost 80% had problems in the neonatal period, sort of medical problems. And then let's see, as I mentioned, they had signs of LBSL as early from birth, sometimes developing as late as 3 months. There were 67% that didn't develop any major milestones at the time they were looked at for the study. And the rest of those all had delayed motor milestones. None of them were able to do unsupported walking. And this provoking factors, that's getting to the fact that, you know, as you know, sometimes with LBSL, if there's a fever or an illness or concussion, things can get temporarily worse, you know, with walking or fuzzy cognition, et cetera. So I don't know what to make of this. None of those patients actually got worse with the provocations. For group 2, though, all of them did get worse with those provoking factors. And so again, some of these kids, it's a much more severe picture than we usually see in LBSL, with in group 1, especially requiring tube feeding, unsupported walking. Christina had asked about seizures. 80% of those patients had seizures. So these are really severe forms. Okay, so that's kind of the summary of sort of where we are with literature and understanding more. And so in terms of what we're doing at Kennedy Krieger, you know, we're trying to understand overall which factors determine rate of disease progression. So it looks like age of onset is a big one. You know, especially that case series I just presented kind of gives us some evidence that the genotype or the type of mutation can also be important in influencing the clinical picture. And that just needs to be investigated more as we build our numbers. And then, and also some evidence that the white matter lesion burden or the degree of atrophy can also, you know, impact at least the severity at the time we're looking. It's just hard to know how much that affects overall prognosis at this point. Just it requires more investigation. So at this point, as Christina mentioned earlier, we don't have any FDA approved treatments, although, you know, we're actively working on potential therapeutic compounds in the lab and in collaboration with other investigators. And so part of my job with a natural history study is to develop outcome measures that represent like clinically meaningful change. So we want to be able to like capture, to quantify disease progression or change in LBSL. So, you know, the hope is that if we see some small change that occurs over a year or two, for example, we want to make sure that once the treatment is initiated, that it reverses that or changes that. So that's part of why we do the research with the wearables. Okay. So yeah, so I already sort of mentioned what the goals are. And so our approach is to get information about gait and balance using wearable sensors and a standardized clinical ataxia scale. We just collect, you know, as you know, a lot of clinical history and exam information at these visits, and also information on cognitive abilities, you know, learning, you know, things that affect school or work performance as well, to try to get a sense of like what's meaningful to patients. So we're doing assessments during virtual visits at participants' homes, and that's been really successful over the last few years to just help increase participation and decrease burden of travel. And I'll say, like, you know, it's small, there's 16 people in the study, but people are like extremely diligent. You know, our retention has been perfect with one exception. And then, and usually people are great about showing up every 6 months. So we really appreciate that consistency. And then we're testing at least at baseline, both LBSL patients and age and sex matched healthy volunteers. And I'll talk later about why that's important. So repeating assessments every 6 months over 5 years are people who've been doing this the longest, or like 3.5 years in. Okay. All right. So in terms of a little bit about what things look like, I think I'll just pop up some more of the pictures here. So we use the Opal system of wearable triaxial accelerometers. So, I mean, these basically look like a Fitbit, but they're just really important for recording movement in a real world context, like the kitchen, the living room. And they've also been used in other neurologic disorders. So as I mentioned, it looks like a watch, and they send a signal to a central sensor through low power radio frequency waves, and we put them on the feet and the waist to get information for a brief series of walking and balance tests. As I mentioned, the lower body is more affected in LBSL, so that's why we're focusing there. And so then for the visits, we send this kit, the Opals kit, a web camera that's remote controlled, a laptop, and a device manual. And this is just sort of a demonstration of actually Ellie McGinn and Beth McGinn at their home with working with the equipment. So as I mentioned, we also use a standardized clinical ataxia rating scale, the SARA, and it's pretty much just a quantified version of the neurologic exam. So it's 8 tests that we do all the time in clinic. It's been used in other disorders that have ataxia. It's a very reliable test. And so one limitation though, is that it's good at showing severity and progression over time, but it's just not great at capturing all ataxia symptoms reliably in early disease. So we're using the SARA and also the wearables to sort of track things in LBSL. And at least in this other ataxia, spinocerebellar ataxia, it looked like the wearables was superior to the SARA and being able to track differences in the early stages. Okay. So as I mentioned, we have 16 people who've been participating in our natural history study, and just wanted to give a snapshot of what things look like for them clinically. So this is a young group. The mean age is 11 years. And our youngest participant joined at 9 months of age and our oldest at 36 months. The age of symptom onset, it tends to be is around 3 years on average, with earliest onset at 2 months and latest at 16 years. I don't know how to explain why we have a lot more boys. We just do. And then in terms of, so the GMFCS is like an overall gross motor function score, and from a scale from 1 to 5, with 5 being very severe, essentially not able to do anything independently. So the mean GMFCS was 1.9. Usually the kids have a 1 or 2. And even though there's some exceptions. And then the max is like an overall assessment of fine motor ability. And so there's also some deficits there. And most of the scores are 1 to 2, even though we have people as severe as a 5. The SARA ataxia rating scale is graded from 0 to 40, with 40 being the most severe. And so well, I forgot to give the range, but I think we've had somebody who entered the study with a score of 0 and has gone as high as I think 39 but this also, the SARA you can use for people who aren't walking as well. It's just the score really jumps once people lose the ability to walk independently. So 75% of our participants are still walking independently, which is great. 1 with a walking aid and 3 are not walking. And I realized sort of looking at all these numbers, so orthotics or, needing something like braces, AFOs, that's actually relatively common. Almost 2/3 of our patients have needed those. And then motor crises. So I mentioned the provoking factors is what it was called in that other table, but we realize that's also very common for our group. And then just anybody, in case anybody's curious about 2/3 of our patients are taking the mitochondrial cocktail consistently. Okay, any questions about this? I probably should have paused earlier. Okay. Okay. And then, something that I think probably is important, especially for parents of younger kids, is what's happening in terms of development in school. So it was very common, like most patients experienced gross motor delays and fine motor delays, about 70%. Speech delays were also common. And then writing problems, almost all patients have experienced that at some point. And then learning disability in about 1/3. Other cognitive means not a learning disability, but something like ADHD or slow processing speed, and 1 case autism. So that's almost 25%. And mostly relating to the writing issues and the learning disability and the developmental delays. Most of the participants, either currently in school or in the past needed formal school support through like an individualized education plan or a 504. Okay. So, all right. So I will now kind of highlight some of the data that was just published in April 2022, just looking at cross-sectionally at our natural history study results. And that primarily highlights the work that we've done with the wearables during the remote visits. Okay. Hopefully I didn't test these videos before, so hopefully it'll work. All right, so for the tests at home, so you can see this young person is in her kitchen. So we've been able to set up a 22-foot walkway in the home, just, in a clear space, kitchen, living room, hallway, whatever it takes. And we do 2 trials of 2 minutes of walking. So they pretty much just walk back and forth, open-ended between 2 points. And then the opals collect this graph is really small, but the point is that the opals get all of this information about every phase in walking, from sort of the foot first stepping off until that same foot touching the ground again. So that's 16 different variables that it collects, which capture every phase of the gait cycle. And then let's see, hoping this will work. Yep, there we go. So this is a 10-year-old girl. She has a SARA score of 14, just for reference. And so she sort of has some hesitation and stumbling with turns. And we actually, I was a little bit worried that, early on that maybe people were looking at that line. So we removed having that tape line in the middle, but it didn't matter. That's still just the tendency. I think it's to make sure that you're aware of where you're walking. We see that very commonly. Okay. So then in terms of kind of what we found, so there's a lot of variability. I think that's one takeaway. And let me orient real quick. So the open white circles are always the age and sex matched controls, and the black circles are LBSL. And so each dot represents one person's performance on the test. So this is stride length. So this is basically just the forward distance traveled by the foot during a gait cycle. So the time from, the foot, let's say the right foot initially making contact with the ground until the right foot makes contact again. And so we can see there's not a huge difference, but you can see a difference here with a lower stride or shorter stride length for the LBSL participants. And that's like what we would expect. So in ataxia, when there's unsteady gait, sometimes the compensation is to shorten the steps. And so, yeah, so that's what we saw. And then for gait cycle duration, that's sort of basically how long does it take to complete that step? Not too much of a difference there. And then for the stride velocity, so the last thing is just stride velocity, and a good amount of variability there. And this wasn't statistically significant, but basically we know from the literature that a difference of about 0.1 meters per second is clinically meaningful. So we saw that here, or 0.09 meters per second difference. So there's a little bit slower walking, from in the beginning with this cohort of people. And so then the next test, that's something actually pretty unique to the wearable sensors. This is not even something that we get like in the motion lab on something like a gait mat, but it essentially allows us to look at step variability. And so if you think about 3 consecutive foot placements made by the same foot, ordinarily that's going to connect in a very straight line, but for LBSL patients, they're unsteady, they're stumbling. Sometimes the gait is a little bit broader based, and so there's just basically more deviations from that expected straight line. And so you can see for the controls, with one exception, I know this was a younger patient, there's really a sort of a tight aggregation of the numbers here around 3.7. And so LBSL patients had much more variability, and and sort of on average a couple centimeters higher LSV, and that was statistically significant. So then for the walking test, so we look at circumduction, which is sort of the lateral swing max, or how much the step swings out during forward stride. And a little bit of that is normal. There's some exceptions, like when there's spasticity in the legs or tightness, sometimes there'll be more of a swing out, or if there's weakness, sometimes we see a little bit more of a swing out. And so there was not much of a difference there for LBSL, but just much more variability. And then we did see for the next one, elevation in mid swing. So that's like the height of the foot relative to its position while standing. And so the LBSL group had on average a higher elevation of the step at mid swing. So that's also called a high steppage gait. And we see that in other disorders where there's cerebellar ataxia or, problems in the peripheral nerves or spinal cord lesions. So it's basically a high step to clear the floor during forward stride, so there's not a trip and a fall. Okay, and so then we looked at turning as well. Yeah, so we so far are not seeing any statistically significant differences there, but it's just something we're following that's important, and where kind of the trend is we continue to see more variability in LBSL and actually a trend towards a slower turn velocity. Okay, so that was a lot. Any questions about that? Okay, great. And then the balance conditions, which, it's challenging for our younger patients, but, we, are getting good data here. So basically we have feet apart, eyes open, feet apart, eyes closed, feet together, eyes open, and feet together, eyes closed. And so those will basically challenge the participant. So removing visual input especially challenges, proprioception ability, or the ability to realize where you are in space. And that's, a deficit in LBSL because of dorsal column changes. And then putting the feet together or narrowing the base of support challenges cerebellar function. So we sort of look at those things separately, and it's 2 trials of 30 seconds each. And so, yeah, so here are, I'll just show you briefly sort of performance here with feet apart, eyes open. I know that's small, but hopefully you can see there's a little bit of sway there. Feet apart, eyes closed. Feet together, eyes open. And feet together, eyes closed, which is very difficult. And so the squiggles above, basically this is the age matched and sex matched control. So her tracing looking from as though from the top down compared to this young woman. And you can see, especially in the eyes closed condition, that there's a very big difference in the extent of sway during those tests. And so then for the whole group, we averaged, so same thing, right? So the white open circles are the age and sex matched controls, and black is LBSL. And we just saw significant differences in all of those conditions, but you can see that there's the most differences in the eyes closed conditions. So that was kind of a really consistent finding. And then one other way to look at this is the Romberg quotient. And so this is looking at, it's just math, the sway area with the eyes closed divided by the sway area with the eyes open. And so normally that's around 1 to 2. And so we saw, quite a big difference, quite an increase in the Romberg quotient in LBSL. So it's just, overall, what it looks like the sway tests are sort of showing us is that the dorsal column dysfunction is very important. That we see it early, especially in this young cohort of kids. And so that's something that we're continuing to follow. Okay. And then I had talked about the SARA test. I won't focus on that a lot, but the point is that this is, a well-used clinically standardized scale. And we saw that lateral step variability measure was very tightly correlated with the overall SARA scores for our patients. So that was, a correlation degree of 0.84, and that was statistically significant in 0.83 if we looked at the total sway area with the feet together and eyes closed in comparison with the SARA score. So it just kind of helping to demonstrate, like the wearables are numbers, but it just kind of helps us it helps confirm that we're on the right direction in terms of clinical meaningfulness. So in summary, our participants walk and turn slower. They've got shorter stride length. They show greater gait variability compared with healthy volunteers. Removing visual input does seem to have a greater impact on the sway area than narrowing the stance space, suggesting that dorsal column dysfunction is important. And then these measures with the wearables correlate well with this clinical ataxia scale. So just a couple points, just kind of highlighting some of the challenges of doing this study in kids. So first of all, like walking in kids, walking in balance measures, it's more recent that they're being systematically studied and that they're being studied using the wearables. We are lucky that there's actually a group at Rush University in Chicago that put out 2 really good papers. since 2021, giving us basically good normative references for gait and for sway in kids. So this table, they had looked at, I think it was about 150 participants ages 5 to 30 years of age. And so what they found, I just kind of want to highlight that the stride length is something that these numbers are small. So I'll just try to, this goes from age 5 to 6 to 22 to 30. And you can see that over time, it changes. So the stride length it makes sense as people get taller, their legs get longer, and the stride length increases from 0.99 to 1.42 over time. The gait speed is also something that increases, in part probably because it's related to the stride length from 1.1 to almost 1.4 at age 30. The cadence and the steps per minute, so it's sort of the opposite phenomenon, right? So smaller people, shorter people take, these shorter steps. And so the steps per minute go from about 140 at age 5 to 6 down to 115. So that's just something that we have to sort of control for and contend with as we're interpreting what's changing with the LBSL, especially our youngest participants, that this is a dynamic process. Okay. So, so despite that, without any correction, sort of what we see is at 2.5 years. So this is not all of our patients, but it's the ones who have been sort of walking from the very beginning. So we see the gait speed at baseline or 0 years. LBSL is orange. Starts out at a lower level than the controls, as I demonstrated with the previous graphs. And then over 2.5 years, we should expect that the gait speed should increase, but it actually did decrease slightly for our LBSL cohort. The double support time, that's the amount of time that both feet are on the ground during the gait cycle. And that tends to be a higher number if there's more instability. And so we saw the double support actually increase somewhat for LBSL patients, whereas it decreases for controls, in stride length, both actually increased. So that makes sense as they're aging over that 2.5 years, but to a less extent with LBSL compared to controls. And then the cadence decreased, but that decrease was a little bit steeper in LBSL. So these are just, things that will be continuing measures from the OPALs that will continue to follow. And it's also an advantage of having more adult patients in our cohort that they're collecting with the Amsterdam group is that, that's one less thing to control for, because things should really remain relatively stable throughout adulthood with that. And then this graph, it's pretty much the same ones that I showed before, but breaking it up by the trajectories of individual patients. And it's a little bit busy, but the point is that I made the warm colors are, or the patients that are having basically changes that would be more consistent with potential progression of the walking variables. So decreases in gait speed, increases in double support, bigger declines in the cadence, and decreases in stride length. And so kind of looking carefully at this, there's a group that seems to be relatively stable and several that sort of their measures consistently sort of change in a way that is more consistent with progression. So, but again, this is a small group. And so we're sort of waiting for catch up for newer walkers, basically, that are part of the study to increase our numbers. So then in terms of the sway area, it's a similar challenge, as I mentioned, and thankfully the Rush group, it also looked at a bunch of young, healthy people and their sway parameters. So what you can see is especially between ages 5 and 9, when standing with the eyes open, this just improves. It's like with neuromaturation, they just get a lot better at it. And especially with eyes closed conditions, they get a lot better over that same period of time. And then it kind of stabilizes out in later childhood, early adulthood. So, so because of that context, not unsurprisingly, we still did see between 0 and 2.5 years, consistent differences between controls and LBSL participants. However, we didn't see that the sway was increasing in LBSL, either for eyes open or eyes closed. So, so essentially, in further iterations, it's going to be really important to consider, controlling for age and height. and we have been collecting height information. So, so next steps are to, basically just continue what we're doing over time with our year to 3 to 5 studies, and waiting again for those later participants to catch up. And then we are working with Dr. England and Hemo, who are here at VMC Amsterdam recruiting LBSL patients. So I think there's 6 that they already have, 6 adult patients that they've been testing. And they've also had MRIs. And as I mentioned, there's also a new partnership with Dr. Henna Tanisma and Emil Jaakallio at the University of Helsinki. And their IRB is pending now, but hopefully any day we'll be able to get started. Okay. And so just a couple of other aspects of what we're doing is, this is a kind of a relatively new aspect of the project, is advanced imaging of the brain and spinal cord. This is just sort of showing again how, if you can see this, this is T2 hyperintensity throughout this whole spinal cord in LBSL. And I've already sort of demonstrated from the wearable study that the spinal cord is important. So we did try just taking a first stab with like all the MRIs that patients have sent us to see if there's any relationship with the ataxia score. And we basically modified a score that's used in ALD for LBSL, sort of expanding. We worked with a neuroradiologist to expand some areas of the cerebellum and brainstem that are important in LBSL, and tried to do a severity score. And so one caveat though is that's just based on the brain, cerebellum, and brainstem. It doesn't consider the spinal cord at all. And so we didn't see a relationship essentially. And so what we're doing next is to do these advanced MRI sequences of the brain, and more specifically the spinal cord, to better understand how changes in the white matter tracts and LBSL are contributing to the gait and balance problems that we see with the wearables. Okay. And so there's several sequences. I won't go into too much detail, but this is based on a longstanding collaboration between Dr. Ali Fatimi and also actually a physician at a physicist and MRI expert in white matter at Vanderbilt, Dr. Seth Smith. And so the 3 that we're working on is, so first of all, this high resolution anatomic imaging called MMFE. And this is like a little bit small, but I think you probably still see the butterfly appearance of the gray matter in the spinal cord. And so this test really kind of helps us to very clearly delineate the white matter from the gray matter. And that improved contrast will just really help us to measure or to quantify atrophy or damage in the spinal cord better. And so that's this sequence compared to T2. The DTI, that's a scan that's been around for a while, but that gives us a better look at how intact the nerve cells are by measuring how easily water diffuses within the cell. And then the last one, quantitative magnetization transfer, or QMT, uses radio frequency to target myelin, and that it's just going to give us a better sense of the quality of the myelin sheaths around the nerve cells. So the nice thing about all of these is it allows us to attach a number, sort of like what we're doing with the wearables, is to sort of quantify what the differences is. So that'll help us with severity. And then we hope also to really capture changes over time. And so we've had, actually this is now 6 LBSL patients that were scanned in Amsterdam so far, and 3 at Kennedy, and we're anticipating more at the conference. So hopefully in a few months, be able to present some data there. So I think that's probably good for now. And I've spent a lot of time talking, but are there any questions? Okay. All right. I don't know if anybody's in the Zoom room that would have a question on the chat. Okay, great. Okay. All right, Christina, I think it's your turn. There's several adapters. Do you have any other adapters?
1 second here. Let's see. I had it a second ago. No, I tried to do presenter mode in case I have a couple slides and now it's working. Okay. Okay, thank you. Just again, I'm Christina Nemeth-Mertz, a researcher at Kennedy Krieger, working on the basic science aspect of LBSL. So I prepared a lot of slides today because I wasn't too sure what our crowd would be. And I really want this to be a session for you. So, you know, if anybody has specific questions or has something that they want to know more about, I'm happy to talk about that. I'm happy to fly over a lot of the slides that I have. I really just want it to be sort of all encompassing. So with that said, I am going to skip over a couple because Amina very nicely just presented a lot of the clinical data, so we don't really need to see that again or really understand the disease trajectory. So just so that we're all on the same page, I wanted to just sort of touch on the very sort of basic science that a lot of this is based on, and that an understanding of would really help to understand the later things I'm going to talk about. So if you were at the scientific review this morning, some of this was touched on, but just so that everyone's on the same page, we all have DNA. That's the basic sequence that make humans human and make everyone unique. So DNA is in the nucleus of the cell. And it's really the recipe that we like to talk about for making proteins and making everything that your body needs to function. So unfortunately, DNA is not easily understood by the machinery that makes proteins. So we have to go through sort of a conversion process to get there. And that's called transcription. And so transcription really just turns DNA into RNA. RNA goes out into the cell and can be made into proteins there. And that process is called translation. And so I'm going to get a little bit more into detail about translation, but basically the final product is to make proteins. And again, proteins are sort of the essence of everything. So working a little bit backwards here, this is a protein sort of in a very simplified model, and proteins are simply made of a polypeptide chain. So the sort of 3D, you can't tell here, but really proteins have a very complex 3D structure, and a lot of that determines how they function. So if that structure is altered, it might affect their ability to interact with other things and really affect their function. So if you unravel this 3D structure, you get a polypeptide chain, and a polypeptide chain is simply made of amino acids. And amino acids are all unique, and so similar to your DNA sequence and your RNA sequence, there's a protein sequence. And so each protein has a very specific order of amino acids that go into making it, which will eventually lead to that 3D structure. And amino acids are coded for in the mRNA by that sequence of mRNA. So as you probably remember from biology a long time ago, they're read in sets of 3. So this is a codon, and a codon will then code for a specific amino acid. And so this very nice table shows you what each of these sequences, I'm going to stick this thing in here, pointer. So if we look at the sequence here, we have, it's not working, GAU, and then you consult that on your table and you see that GAU leads to an aspartic acid, which is a D. And then we have some machinery that comes in with that D, and then that D eventually joins the growing polypeptide chain. And so that's just something that goes over and over and over again, and it happens really fast, and it happens millions of times. And so this is where we get to the tRNA synthetases. So this is like DARS2, and it has a very specific and very important role for making that whole process happen. So DARS2 is really the enzyme that joins that tRNA to the amino acid. And then that amino acid joins together, the DARS2 goes away, and then that tRNA goes and does its job in the growing polypeptide chain. So what's unique about DARS2 is that happens in the mitochondria. And so this is a process, the protein translation process, and the protein machinery is something that's present in the cell, but it also has a sort of duplicate set that occurs in the mitochondria. So the machinery that makes proteins here are unique from the machinery that makes proteins in the cell. And what's interesting is that DARS2 is one of many. So there are 19 different tRNA synthetases that function in the mitochondria. DARS2 is one of them. And then there's 20 that function in the cytosol, so in the cell body. And then there's actually one of these that functions in both places. And so DARS2 is unique in that it carries the aspartic acid, but it's not unique in that there are several other enzymes that are just like it that do the same thing, serving a different amino acid. And what's really unique, especially and very interesting to me, is that mutations in any one of these genes results in a very different disease, basically. So we have DARS2 here, which we know obviously affects the nervous system, but if you have a mutation in YARS2, you have a dysfunction of skeletal muscle. So it's really interesting that these enzymes that are so similar and that perform a very similar role can result in a wide array of different diseases. And if you break down just the nervous system disorders, so now just looking at the neurological conditions, we do see a lot of variability there as well. So some cause of peripheral neuropathy, some are specifically epilepsy, some are associated specifically with intellectual disability. So it is very interesting and very, very complex, basically. Any questions there? And one more thing I'll just point out here, I believe LBSL is the most prevalent of all of these disorders. So, and as you know, that population is very small. So the population for all of these others is much smaller. And this is just simply a slide that reviews all of that. And this is a slide also that Amina had, just demonstrating in 2014 many of the mutations that were associated with LBSL. And as you can see, the blue sort of coding regions here are the exons. So there's many different mutations associated with exon mutations, but then many, and then the most prevalent, as we said, up to 95% of patients have an intronic mutation. So in that sort of non-coding region, and that seems to have a pretty significant effect on the protein. So my job really focuses on modeling diseases, and modeling diseases, of course, is very important because you have much more flexibility to understand the disease, and it's something that's important to test therapeutics and to really move forward in sort of driving a mechanism from disease. So there's multiple different ways to model diseases, and they're models, so they're not perfect. They'll never be perfect. We will never test on people, these sorts of things. So your phone is up here. And so we really need a way to understand these diseases. And so our plan and our goal is to have multiple different models, because not one of them is perfect. So if we have many and we can get sort of a consensus across these models, that's really our best bet at understanding the bigger picture. And of course, there's pros and cons to each. And so what we focus on is mouse models and cell models. And just very quickly, the pros of mouse models, of course, is that they're well established. There are decades more of research understanding mouse behavior, understanding mouse physiology. We know sort of the basis of these mice. The mice that we use in the lab are very well characterized. And so if we introduce a genetic mutation into those mice, we have a pretty standard set of ways to characterize the changes and to understand what's going on. And then, of course, it's an entire system. So if you change something in the brain and you see an effect in the digestive tract, that's something that we would understand in a mice that you wouldn't be able to understand in a cell model, for example. They're optimal for therapeutic testing for the same reasons. And then you can very easily make genetic mutations in them. So there are companies that do this. You can order a mutation in any gene and they'll send it to you for a price, of course. The cons, we can't replicate all types of mutations. That mutation that's so common in LBSL in that intron region, we cannot replicate that in a mouse. That's just the mice, their genes aren't set up exactly the same way as humans, so we can't generate that same splice site mutation. So that would be a con. Mice don't always develop a disease phenotype. That's a huge problem for ALD. The ALD mouse, we cannot get a cerebral phenotype in those animals, and that's just the way it is. Experiments can be very long. With the LBSL mice, that's something that we see. It generally takes about 9 months for them to develop a very significant phenotype. If we're testing a therapy, that's a long time to wait to see if that therapy had an effect. And then they can be expensive. Of course, we have to feed these mice, we have to house these mice. We have very strict regulations on how these mice are kept, and so that costs a lot of money, but it's certainly well worth the cost. For the cells, the pros there, we can have very patient-specific cells. So we have cells from our patients, and sometimes we can see correlations between the way that the patient performs and the way the cells perform, and that's a really nice thing to be able to understand. We can look at cell type specific effects. LBSL affects the brain. We really want to look at brain cells, and that's just really something that you can do easily with cells. We can basically derive any cell type we want within with some exceptions, but it makes the most sense to study the cell types that are the problem, and that's what we can do. They respond to therapies, and I'll show you some data on that. And compared to the mice, the experiments are a little bit shorter, so that's certainly a pro. The cons, they take time to optimize ideal conditions. You can get a protocol from another lab, but cells may not behave in my hands the way that it behaves in someone else's hands, and so it always takes time to get that sort of protocol down. Cells, of course, grow in a dish in an incubator, and that is very different from the way cells are in a body and a brain. And we have to make up for those changes, or we have to make up for those conditions, and sort of understand that the changes that we observe in the lab might not be what we see in the patient. And then, of course, they're expensive. So growing a neuron in the lab takes very specific media, takes very specific small molecules and temperatures and equipment. So a lot of that stuff adds up very quickly. So I'm going to skip this slide. We talked a little bit about the pros of mouse work. So I'm just going to very briefly, I don't know if you guys are interested in hearing about the mouse model. I can skip to the cell work, or we can touch on sort of what's new. Okay, so I'm going to skip a little bit. This is some work from a collaborator's lab in Cologne, Germany, and they designed this DARS2 mouse. They knocked out DARS2 in the brain, in the skeletal muscle and in the heart, and observed some changes. And so we were able to get these mice from that lab and sort of create our own mice. And this was published in 2020. So I won't spend too much time going over this again. Basically, what we learned from this is that DARS2 is essential for life. We cannot have a mouse that lacks DARS2 completely. And so I'll touch on this slide a little bit just to talk about some new things that we have coming up. But when we first started this, we tried to delete DARS2 from nestin cells. So these are cells that, this thing does not want to work. So nestin cells are basically neural stem cells, which is going to be this one up at the top. So these cells would differentiate to be all sorts of cells within the brain. And if you knock out DARS2 within those cells, it's not compatible with life. So we need DARS2 in these neural stem cells. It's really too early of a process, or too early of a cell type to knock that gene out. And so we then went on to PDGFR alpha. So this is an oligodendrocyte precursor cell. So now we're just sort of affecting these types of cells. And these animals didn't really have a phenotype. So deleting DARS2 from those cells resulted in basically nothing. The cells, or the animals, looked quite normal. When we did a similar thing with KMK2 expressing, so these are a specific type of excitatory neuron, then these mice had a very significant phenotype, and that's the data that was published. So just to sort of touch on this, in the lab now, we're actually looking at two other types of mice. So one is a OLIG2, so that's also a different type of oligodendrocyte precursor cell. So that's something that we're sort of working on generating now. And the reason for that is the method that is used to generate the PDGFR alpha cells is actually a little bit more complex than the KMK2. So the animals require a drug to be on board at the same time for that gene deletion to occur. And that's something that's pretty hard to time. The drug is sort of short lasting, and so it requires multiple doses. We don't really know if it's on board or not. And then also the timing, the age of the animals when they get the drug and when the cells are modified is really quite variable. So it's a hard thing to monitor, and it's a hard thing to know that you get right. If we're looking for a subtle phenotype, it's also close to impossible to really know that we're doing it properly. So, is there no phenotype, or is the drug not on board? So the new mice that we have now, OLIG2, so this is something that the mice are born with, and we don't have to worry about having that drug on board. And it's going to be sort of like the KMK2, where we know that deletion is made automatically when the cells are generated. So that's going to be a little bit more of a surefire way to know if this oligodendrocyte lineage is more subtle than the neuronal lineage. And then the other model that we're working on is is a dorsal root ganglion. So this is a specific population of neurons that feed into the spinal cord. So these are sensory neurons, and this is going to be the most sort of closely related to LBSL patients. So we know that LBSL affects the spinal cord. The mice that we published on, the KMK2 mice, that is selectively affecting the brain, which is sort of what we see in the very early onset, the very severe cases, but it doesn't represent the vast majority of LBSL patients. So this dorsal root ganglion model should be a little bit more translatable or a little bit more accurate to reflect LBSL patients. So we're going to see if affecting these cells within the mouse spinal cord also has sort of gate consequences, essentially. So this is the data that was published. What we know about these KMK2, so this the brain neuronal model, is that these animals have a very significant phenotype, so they get very hyperactive. So this is just showing that by 9 months of age, these animals are essentially running around the cage crazy. And this is also correlated, or this happens at the same time as significant atrophy in the brain. So when the animals are about 6 months of age, they're starting to show, compared to controls, a reduction in the brain area. By 9 months of age, you can see that this brain is much smaller than this brain, and that's reflected in these MRIs too. So all of this white space here is just atrophy. That's just basically fluid and a lot of empty space in the brain. So there's significant neuronal loss in these animals. And we can see that if we take the brains and we section them. So this is if you imagine a brain and you take basically a bread slice, a vertical cut through the brain, then you're going to see a section like this. And you can appreciate the difference here between the thick cortex here and the very thin cortex here. And what we also saw in these animals is neuroinflammation. So the cells right here are kind of round. We call them amoeboid, as opposed to these sort of more ramified. So this is what a healthy microglia or inflammatory cell looks like. When they get activated and they get angry, they sort of take this phenotype. And that just means that there's a lot of inflammation in the brain. And that's pointing out an inflammatory marker. So just to summarize sort of the trajectory of these mice, we know that they develop normally. We know by 6 months of age, we can start to see this behavioral change in the cage. We can also see that their brains are starting to atrophy. And by 9 months of age, we know that that's sort of very significant neuroinflammation and very significant neuronal loss at that point. So our question is sort of, when does this happen and sort of what's happening in this sort of middle region? What's precipitating that? And so what we did is we took these animals and isolated the affected parts of the brain. So where all that neuronal loss happens, we cut that out, sent it off to a company and did something called RNA-seq. So we took that RNA, if you remember the central dogma in the beginning, basically looking at the middle section, the RNA that is the recipe for making proteins. And that's just a way for us to understand what genes are active and what the body is producing or what the brain is producing, what genes they think are important. And what we saw is that at 14 weeks of age, there was a significant number of significant number of gene transcripts that are downregulated. And so that's kind of what this peak is on the left. So anything on the left of this center line is downregulation, and anything that's over here is sort of upregulated, or we're making more of those genes. And anything that's green is considered to be significant. And what we saw is basically a lot of these genes, as we can expect based on the histology that we looked at, are related to inflammation. So that we know that there's a lot of inflammation, and then there's a lot of cell stress and a lot of processes that are suggesting kind of cell death. And that's reflected also in here, which is a little bit more of a colorful picture, showing you that all of these red genes are increased, and these are all related to inflammation. And then these genes in the LBSL mice that are decreased are all related to neuronal processes that are no longer happening because we have significant neuronal death. And one more sort of picture of the same thing, showing increases. So the brown is all increases in inflammatory genes, and then very specific processes in neurons as well as oligodendrocytes that are decreased. And so here we can see the specific genes that are either upregulated or downregulated. And based on this data, what we did is we treated these mice with a pretty potent anti-inflammatory, antioxidant drug. We did it very early on before any of these processes happened. So if we go back to that timeline real quick, right here. So we know that by 14 weeks of age is when we start to see that increased inflammation. And so we treated these mice starting very young and it had no effect. So the trajectory was exactly the same. And that either means that this is a very powerful process or our pretty potent anti-inflammatory antioxidant is not really targeting the same thing or the right thing. So let's go back over here. With that said, the inflammation that we see in the mice is not something that we think is totally reflected in the patients. So that's not something that is reported or even observed. So, you know, it could be something certainly related to the neuronal death that we're seeing in these animals, but we don't think it's a very important target to be following in these mice. So looking now on the cell work, what we use is induced pluripotent stem cells. And what we do is we get these from the patient's blood. So we take the blood, we send them off to a company who converts these blood cells into stem cells. And these are cells that are capable of being basically turned into any other cell type. And so we work with Dr. Mingyao Ying. He's a, yes. So yeah, two separate models. So we did Cre-Lox, basically recombination. And so we had one set of the CAMK2 mice, that's the data that was here. And then if we did the Cre-Lox recombination in PGGRF, these are tamoxifen inducible mice, then we didn't see a phenotype at all. So it's a neuronal disease, we believe, or, you know, oligodendrocytes. So we needed to, if we couldn't do Nestin to affect the entire brain, we needed to basically target specific populations of neurons. CAMK2 is one of the sort of the more common Cre models that are available. And so that was sort of the first target. So hippocampus, prefrontal cortex type thing. These are sort of easy cell types or easy phenotypes to characterize with those mice. It was basically targeting some sort of neuronal population and then some sort of oligodendrocyte population. Collaborators in Germany did a similar thing. So they also did CAMK2 and then they did PLP for oligodendrocytes. And they also found that PLP mice showed no phenotype while the CAMK2 had a significant sort of phenotype. It's basically the easiest option. The DRG model is the next one to go after a different type. The same group in Cologne also did a cerebellar knockout, and they had a significant phenotype there too. So it's what's available pretty much. So with the iPSCs, this is just a general schematic of kind of what you can do with them, taking them into a dish, treating them with reprogramming factors and small molecules to sort of push their differentiation into any sort of cell type that you're interested in studying. We, of course, are interested in nerve cells, and what we did was motor neurons. And so along with the help of Dr. Ying, we pushed our patient cells using NGN2, neurogenin2, into this motor neuron stage. And this takes about 14 days to do. And as you can see in this video, if we image these cells every 2 hours for a period of 24 hours, we can see the development of these cells. So neurons are very complex. They have a very nice arborization. They grow. They have dendrites that reach out and communicate with other cells. And you can see that they form these very complex networks. And that's something that we can monitor across our different patient lines. Excuse me. So this is data showing sort of very baseline. This is just taking the area of the cells. So the amount of space that these cells take up over a 24-hour period, with the green being the control. You can see 3 different patients here of LBSL cells over, I guess we're looking at 1200 minutes here. You can see that many of these LBSL cells don't differentiate or don't grow, don't expand to the same extent that the control cells do. So we have decreased sort of complexity and decreased arborization of those LBSL cells. And if we look a little bit more specifically at 2 factors, so this is branching. So this is the ability of these cells to sort of form these little legs that come out. So that would be branch points where they sort of separate like a tree. And then also looking at the neurite outgrowth. So the length of those outgrowths, we can see that LBSL is consistently decreased compared to the control cells. So they just fail to have that complex arborization that's important for neuronal communication. And I think you can see that reflected in the 2 images here. Neurons are unique in that they're electrically excitable, so they fire action potentials, and that's how they can communicate signals to other cells. We can measure that by plating these cells in a specific cell or a very unique plate that has electrodes at the bottom. And the electrodes will monitor the cell activity. If we put them into a machine that looks like this, we can monitor their mean firing rate over a period of time. We put them in there multiple days, and so we can see sort of how they mature and how they fire over time. So a normal cell would fire, would have a rate in a given time period, say an hour, at this level, so control basically levels. And what we see is the LBSL cells sort of fail to have the same level of electrical activity. And this is going to be important for development. It's going to be important for signaling and that sort of thing. So this is also another important finding that we've seen across multiple cell lines. And finally, looking at mitochondrial activity, this is a test we call the seahorse. And this is a very commonly used paradigm to study mitochondrial activity. What we see here is the control cell profile of what the typical responses are for a seahorse test. And we can look at many different aspects of mitochondrial function, looking at baseline oxygen consumption of the cells, the amount of oxygen consumption that is ATP linked is sort of what we see in that second section with the addition of a drug called oligomyosin. At the third section, we see max respiration. So we treat it with a drug that pushes these cells to the max of their capacity. So the total amount of energy that they can expend. And then finally, we sort of give a drug that sort of knocks it completely out. So one of the very important things that we look at is sort of that spare capacity. So if this is baseline function and this is the max ability of the cells to really exert themselves, we call this the spare capacity. So that's how much energy you have on board if you're all of a sudden chased by a tiger. So that's what you can sort of push your cells to do under extreme circumstances. And if we look at the control compared to LBSL cells, we basically see that with the addition of any of these drugs to push the cells in any direction or the other, there's a diminished ability of those cells to respond. So one of the biggest kind of take homes here is that spare capacity is gone. So they don't have any more energy to give other than what they're giving already. And if we look at multiple other patients, we sort of see across the board that we have a very similar profile of these cells and their ability to perform under stress. We take the same data and look. Did you have a question? No. Yes. Yeah, go ahead. So no, good question. So the question is, is the combination of these patient lines based on anything? And it's not. It's basically we do multiple iterations within a plate, and so there's only so many wells in the plate, and it's the timing of growing the cells is very complicated. So we can do 2 at once. And the idea is to do this many, many times in many different combinations and see if we get sort of the same results. So the ordering of these is not, yeah. And I want to compare them each to their own specific run control. So I didn't combine them into one plot because the controls perform slightly differently every time. Looking at the same data in a little bit different way, just to compare the baseline respiration and the maximal respiration, you can see the differences between sort of the control here and the control here, and then how these LBSL cells perform. And then if we take the difference between those two to get the spare capacity, that's sort of what you're seeing here. So the spare capacity, obviously, for the control is much higher. And then for all of these LBSL patients is diminished. One actually went down even more. So sort of a negative ability to deal. And then looking at ATP production, so the amount of ATP that's through oxidation and then through glycolytic processes. So you can see that these are sort of all the same, and it's that ATP production from the oxidative respiration that's really sort of affected there. And that's where the problem is. Getting a little bit more complex here. So what I've showed you so far is characterizing the function of these cells. And what we really want to understand is the DARS2 protein, and is DARS2 doing its job with protein translation? So we know, of course, that the tRNA synthetases combine the amino acid to the tRNA, which participates in protein translation. So is that job being performed? And if we first looked at the DARS2 protein, what we see in the LBSL patients is baseline levels of protein. LBSL is across the board reduced compared to control. In a control situation, it appears that motor neurons require less DARS2 protein overall. That just seems to be the way it is when the cells differentiate and mature into neurons, less of that DARS2 is required. In both cases, as an iPSC or a motor neuron, LBSL has less protein than control, and you can see that in these Western blots as well, which is the same data, just in a different format. If we look at the total protein of a cell, we did proteomics of LBSL patients compared to control patients, and we see in LBSL that there's an increase in 441 proteins that are sort of made in a greater abundance than control, and then 27 that are downregulated. If we look specifically at DARS2 across the 3 different patients that we saw, since DARS2 adds aspartic acid to polypeptide chains, if a protein contains a lot of aspartic acid, we would assume that that's not actually being joined. And what we see here is the percent of aspartic acid within the protein does not correlate at all to the changes of that protein. So this protein right here, ND2, doesn't contain a single aspartic acid in its protein sequence. However, it is reduced by 50%. So it's not a relationship between the ability of aspartic acid to be a part of that protein, if that makes any sense, I hope. This is another way of looking at similar data. So again, if DARS2 wasn't doing its job, we would expect aspartic acid to not be present, and we would expect maybe a different amino acid to be substituted, and it doesn't seem to be the case. So the amount of substitution found within LBSL proteins compared to control is not different. So we assume that the mitochondria is still able to faithfully create these proteins in a way that's mostly correct. These are other data that are basically showing the same thing, so I'm going to skip over this for right now. So we know that mitochondrial proteins don't seem to be all that affected. So what is changing? We did see that there were significant upregulations and downregulations in that proteome data. So what are the proteins that are changed? And it seems like a lot of them have to do with regulatory processes. So a lot of them, as you can see with the gray bar here, and then also with the orange, a lot of them have to do with protein binding, with DNA binding. So that's a lot of regulatory processes dealing with mRNA and dealing with the ability of these cells to interact, and the ability of these cells to regulate processes specific to making additional proteins. And so that's sort of a molecular function that we were not expecting to see in this context. So one way to better understand a lot of these changes is to do this in sort of a more complex model. So one of the more recent things that we did is we took these iPSCs and we let them sort of differentiate as they would to form sort of a more complex structure. So these are called organoids. We like to refer to them as mini brains. And this is an unguided protocol that one of our medical students in the lab, Schertzy, was working on for a few years. And so you basically take these iPSCs from the patients, you allow them to differentiate on their own. So if you put them into a plate and you allow them to grow, they form sort of a 3D ball, and that 3D ball will develop and differentiate into different cell types. So here we're not really pushing it to become a specific cell type. We allow it to do essentially whatever it wants to, and it forms this 3D ball, and it forms an organization that is somewhat similar to what we would see in a brain. So you actually get layers of different types of neurons, and you can get different types of cell types. And what's really great about this is that in the brain, your cells interact with multiple different cell types, and that's something that's natural. That's not something that we can observe in a neuronal culture of just one cell type. So here we're allowing that sort of complexity to take place, and we can really see what these mutations might do in a more complex structure. What we do at this point is similar to sort of the proteomics approach, is we take these cell balls, we let them get pretty old. We grew them until they were about 70 days old, and then we put them into a single cell solution. So we sort of destroy this ball structure. The cells are still alive, and we put them into a single cell solution, and then we can sequence every single cell independently to see what that cell is doing. The process that we used is a highly specific process. I guess it has very high resolution. So sometimes you can just look at the abundance of different genes. And what we really wanted to do is we really wanted to understand how these genes were being made and how these genes produce transcripts and how those transcripts can produce proteins. And so to do that, you really need to look very closely. And so that's what we call a high resolution approach. Here's a picture of some of these organoids when they're sectioned up. So if you take slices of them and then you can stain, and what we can see is that we're getting multiple different cell types. We can get neurons, we can get oligodendrocytes, and that's something that we're really interested in doing. When you do the single cell sequencing, this is just a distribution of all the different patients. So we did all of our 7 LBSL patients compared to 3 different controls. When you plot them onto this map, where it plots cells of it plots basically on characteristics. So if cells have very similar characteristics, they're going to be plotted next to each other. We see there's a nice distribution of all of our different patients represented. So that basically means that we're not having any sort of biases, and all of these patient cells are sort of behaving similarly, and we can sort of have more confidence in the data. When we look at the different cell types, we got a few different signatures here, a few different cell profiles, and what we're most interested in sort of is the neuronal lineage, such as the neuroepithelial cells, radioglial cells, cortical neurons, and then a little bit of the glial cell development. So we didn't unfortunately pick up a lot of these white matter cells. We didn't pick up the oligodendrocytes that we're hoping to see, but we think we got a decent amount of information from our neuronal cell lines. If you have any questions, feel free to stop me. It's going to get a little bit more complex here in a minute. So one of the things that we really wanted to look at was DARS2. So what is the function of, or what is the structure of DARS2, and how is DARS2 behaving in these different cell types? This is a very simplified version of the gene. Each of these vertical lines here is an exon. So DARS2 has 17 exons. We're going to see 17 vertical lines, and this is what we would call a full-length transcript. So this makes a full, healthy DARS2 protein. There are multiple recognized different transcripts of DARS2. So if you look online in journals and databases, DARS2 is capable of making multiple different proteins. And so what you see here in all of these lines is basically a different combination of those exons. So you can see that in some of these transcripts that make proteins, we are here, you're skipping certain exons. That's okay in some cases. So all of these are proteins that are made and probably serve some function, and we don't really understand them, but they exist, and it's something that the cell requires. Cells also produce a number of transcripts that are not functional. So the red ones make proteins successfully, and these are actually degraded. And one thing that you can notice here is this third exon is missing from all of these. So if the third exon is missing, most of those transcripts get degraded and no protein is made. So that's going to be important in a second. So this again is just the DARS2. That's our 17 different exons there. And if we look at one of the specific cell types, so this is neuroepithelial cells that we picked up in our organoids, what we see in a control cell. So here we're basically looking at the transcripts of specific cell types. So what we see in control cells that are going to be blue is that you have your same exons. So 1, 2, 3, all the way up to 17. And then these lines linking them basically mean that the transcripts are complete, that one exon is connected to another exon. So this is sort of a normal situation. You can see here that this particular transcript skips a little area here. That is normal. That is something that's recognized sort of in the data, in the literature. And that's also what we consider sort of a control situation. Looking at LBSL, all of the sort of orange shaded transcripts are LBSL transcripts from specific neuroepithelial cells. And what you can notice here is we have bands that skip. So many of the transcripts produced by this particular cell have all of the exons, and then some of them are actually skipping exons. And remember, if we're skipping exon 3, that cell is not producing a transcript that makes a full length protein. So this cell would make protein sometimes and would not make protein sometimes. This one is skipping exon 3 all the time. So this cell would never produce a functional DARS2 protein. If we look at radial glia cells, in control situations, you see that this transcript is sort of truncated, which stops at 13. This is something that's picked up in the data, so that's actually still a fully functional protein. LBSL sometimes produces also these truncated transcripts. This one again skips exon 3, which means that cell would never produce a functional DARS2 protein. If we look at cortical neurons, so these are sort of more complex neurons that we're interested in. Here in the control situation, you have some that produce a longer transcript, some with a truncated, shorter transcript. And then if we look at cortical neurons from LBSL patients, every single transcript that's made is again lacking exon 3. So none of these LBSL cells are producing a DARS2 protein. So this is really gets at sort of the problem in that reduction in DARS2 protein that we picked up earlier in those Western blots and some of our other assays, is that when you get these cells that are more differentiated to a neuronal phenotype, these cells fail to produce DARS2 protein for the most part. If we do, if we look at all of the genes that these cells are making and characterize them based on different processes, one thing that we noticed is that, let me see if I have a picture of this, I don't. One thing that we noticed across our 7 patient lines is that they clustered differently. So we naturally found 2 different very well-defined groups of patients. And those are what we call group 1 and group 2. So some subset of patients had a similar profile and another set of patients had a very unique other type of profile. And what we found across these 2 is that there were similarities and there were also significant differences. So both of these groups of patients have differences in RNA splicing. However, group 1 had a downregulation of RNA splicing and group 2 had an upregulation of RNA splicing. And then we also found that group 1 seemed to be more associated with regulation of cell death, and our group 2, thought I had another one. Oh, you can't see. So group 2 has more processes that are associated with mitochondrial function. So you can see oxidative phosphorylation, ATP synthesis, mitochondrial respiratory chain complex assembly. So these cells seem to have more mitochondrial deficits, and these cells seem to be more associated with cell death processes. And if we look at the data or go back through the data, really considering those changes, what we did notice is that the types of mutations that these patients had really determined which group that they were in. So some of our patients have mutations within these splice sites. So these are the intron mutations. They all clustered together. Sorry, these are the splice site mutations here and here. They all clustered together and they're actually associated with group 2. And the patients that have a missense mutation or a mutation within their exons, that's actually the group 1 patients. And again, if we go back to that mitochondrial data and look at those profiles from the seahorse, we see that the group 2 with the oxphos or the mitochondrial function processes within the slides in the last, the data in the last slide, these are the patients that had a worsened seahorse profile, and the patients that had sort of just a diminished but still had a response to a lot of these mitochondrial function drugs are the cells that are associated with the cell death pathways. So it looks like there is some sort of phenotype relationship with the ability of the mitochondria to function and the data that we picked up in the single cell RNA-seq. So that was kind of complicated, but I hope that made some sense. So our goal with both of these models to eventually use them in a translational setting. So as a test bed for therapies. With the mice, we've tested the antioxidant, anti-inflammatory. We've also looked at something else called the integrated stress response, which I didn't touch on here. And then in the cells, we actually also did ISR inhibition and activation. And then one of the things that we're working on now is sort of the antisense oligonucleotides, and then the AAV9, which is the gene replacement that we discussed a little bit earlier. So hopefully, as those projects progress, we'll have some more data on that, and that can be something that we can share soon. So conclusions, we know that DARS2 is essential for life. We know that the conditional deletion in the mature neurons is something that produces a significant phenotype in the mice. It appears that the neuronal phenotypes are much more worse than the oligodendrocyte phenotypes. And this has been confirmed by other labs. And then these mice are going to be essential for testing therapeutics. And we hope these new mouse models that we're developing will add to sort of that strength of data. The cells, the patient neurons show reduced amounts of DARS2 protein. As these cells mature, it seems like the amount of protein goes down in both cases, more so with the LBSL. The patient cells develop normally. We do see abnormalities in branching and outgrowth as they age. We also see reduced firing in the microelectrode array plates, and then of course deficits in the seahorse mitochondrial function. The 3D organoid models, they offer a more in-depth view of the transcript level changes. They're also a really nice model for looking at the complexity of the cellular interactions. And hopefully that's also going to be good for a test bed when we start to test therapeutics. And then of course, they're the cells, the neurons, and then again with the organoid models are good for both developing and considering multiple therapeutic strategies. We can look for penetration of the AAV9s. We can look at ASO function and the like. So much more to do, and hopefully we'll have some new data on that. And then I just have a acknowledgement slide with the team and a lot of people to thank. And then of course, a picture from our LBSL conference in 2018. So any questions?